Thank you, Patricia, for having me, and thank you everyone for being here today into the International Institute of Biosensing. And also hello to everyone online. And please feel free to speak. I can kind of check the chat, but please feel free to just unmute and interrupt me if you'd like to bring anything up throughout the talk. And yeah, so I'm talking a little bit about some of the Air Force research work that was recently funded, and so I'll just to give a little bit of a background. So this is work that is really something that is actually kind of new to me. So my background, as Patricia mentioned, is more in cardiovascular device and surgery, medicine, and better understanding underlying disease etiology and physiology. So that's my background. I've studied a lot about tissue device interfaces and was really brought into this project to bring in that biological, physiological side. So this is it's very fun for me learning a lot more about physics. And Beth, also who's here in person today, her background's in physics, and so I really enjoy talking with her and kind of geeking out about some of the physics aspects of things. And so, but just I'm kind of saying that as a precursor because I'm not a physics expert, and I've been a fantastic collaboration with international collaborators for this project, mostly a US-Italian collaboration, but also really this is a basis that's been built over many years by Professor J.P. Jumping Wang in electrical engineering, and Thomas Brown at Tor Vergata in Italy, and so I'm just it's been really a pleasure to start to work on this. And I'm going to talk a little bit about well, I know this is kind of a long crazy title, but really the overview of what I'd like to talk to you about today is just a little bit about this international project with the Air Force Biophysics program, and also related to neuromorphic engineering. That's a topic that I've been very passionate about, brain-inspired computing and neuromorphic engineering, and how we can use these platforms in device tissue interface, and how we can have our physiology interact with computers in computing in a way for real-time feedback, et cetera. And I want to talk a little bit too about some of the power of multi-scale translational research aspects within this, and how we can leverage AI and computational modeling for precision. And so, the Air Force project, there's a basis for human augmentation in this. This is, again, I'll go back to like this technology platform within the title, but overall what we're trying to do with this platform is to better understand how we process vision and information, our ability to manipulate photoreceptors and other biological tissues with different wavelengths of light, different magnetic fields, how we can potentially be able to detect, sense, see, and so like maybe from an augmentation standpoint, but just better understanding how that processing happens, and the information that we perceive from our periphery. And that's kind of going down a little bit of the rabbit hole of interoception and glioception, which is also a collaboration and work of mine, but I'm not going to really focus on that in this talk. But also there's a lot of really great work being done by collaborators on better understanding glial cells and how they perceive information within our periphery, and that's specifically with Dr. Valentina Benfinati. But that's another rabbit hole too. So, and then just also kind of just wrapping that up to how we have an interaction with this device tissue interface, and like what are things that we can do for moving forward with that. And so, again, this is the title, right? So this is it's a technology platform for photoelectrical and electromagnetic simulation. So this is arrays. This is work that's been developed by Dr. Jianping Wang, and I put this up here because he just recently was awarded the Faculty Innovator of the Year, and was also on Time. He was top inventors of the year this past year. So that's in collaboration with him as the PI of this particular project. And from the electrical engineering and side, and then Thomas Brown. So this is from the CHOES lab, which is outside of the Ring Road in Rome at Tor Vergata. So I had the opportunity to visit their facility earlier this year and to better understand some of the work that they're doing, which is really very interesting. So it's actually the CHOES lab is mostly developing technology around solar panels and better understanding how we can leverage different wavelengths of light and have a very, just like an amazing facility, and working on developing printing biologics on certain filaments in a single cell layer. So that is part of this platform that we are working on doing here. And so the part about understanding neuromorphic vision, it's how can we create this to be a closed loop system? And what is it? So this is, you know, this is not like a huge amount of like result driven things in this talk. I mean, talk that is new, that is the new work that we've been doing, but it's a lot of just to really describe like where we're going with this, and also like some of the preliminary data that we have that has been extremely exciting to share. And again, this is funded by the Air Force biophysics program through Dr. Ben Solomon. Okay, so this is we're kind of capturing a couple of really unique, maybe some of this is emerging technology, but it's just innovative. It's innovation and innovative ways of thinking about leveraging technology, and so capturing the photoelectric electromagnetic stimulus. So we're better understanding leveraging some of these photoelectric fields, and this includes materials. So how can we, so again, this is kind of going back to the CHOES lab, like how do we leverage different types of materials and filaments that can both hold cells live, that can interact with tissue, that can be developed in a filament and so forth. And this is also work that's already been there's been a basis for this that has been published by Dr. Mark Brown and Tor Vergata. And then also looking at quantum sensing. So this is, I mean, the reason why we're bringing in quantum sensing is to better have like really high precision, to be able to better understand entanglement. These are things where we can like start to better understand entanglement. Well, not understanding as much as like capturing that as a tool for a feedback loop, and then we can also be sensing the quantum field. And the really unique aspect of this is that in this platform, we are using non-contact sensing. And so we're doing a combination of electrical stimulation, electromagnetic field stimulation, trying to use different wavelengths of light for stimulus, and then sensing using at a quantum level at a non-contact, and also using spintronic sensing. And so the spintronic sensing is really leveraging this in a quantum spin. So we're like looking at different ways of being able to obtain a magnetic field. And then just like the overall idea of bringing this into a neuromorphic engineering framework. And, you know, so I'm just kind of, I'm basically like unloading the title here just to like give you a heads up. But this is like where we're really trying to capture neuromorphic engineering. And so this is a way that we can really optimize sensory processing. And so that's really like the big thing for the sensory processing is how we're capturing, how just as an overall concept, how we're capturing information through our eyeballs, and how that's being transmitted to our brain, and how that's being processed. And so that is something that from an electrophysiology side, like, so I have my background is in cardiac electrophysiology, so it's been transitioning into better understanding the electrophysiology of the optic nerve, and that intersection, the physiology of the intersection between when the optic nerve is actually interacting with different aspects and physiological aspects of the eyeball itself. And yeah, so again, how we're leveraging the tools and integrating this device tissue interface overall. So this is basically the network that we're trying. This is our trying to demonstrate here. So again, this is, yeah, closed loop. So we're trying to make this a closed loop system. where we can create a situation. So all these things separately have been established. And so now we're trying to integrate this into a platform that can be closed loop. And so in other words, for instance, if we have cells, if we have in vitro cell network that we're evaluating, that we can expose those cells to either different wavelengths of light, to different electrical magnetic stimulations, and then can then use the quantum sensing to better understand that output. And then also like some real in vivo neural tissue and triggering with tunable frequency. So the tunable frequency part is really key here, because so we're using these different parameters and different wavelengths of light. And the idea of the closed loop is that if we know that we can expose, and I mean, just like, okay, from a very basic overall physiological standpoint, like the eyeball with a specific wavelength of light, or specific frequency, and we know that the output will be in a certain quantum, the quantum sensing will have a certain amount of parameters, then we can create precision for the wavelengths of light that we're able to see, and creating the photoreceptors. And the idea here that this is kind of goes back to, I'm going to talk about this a little bit more in a minute here, but the idea of how we can use precision medicine and leveraging biosensing for precision medicine. And I think it's worth just taking a minute to take a little bit of a step back into the overall kind of idea here in terms of what it is that we want to look at, and the scale. And I think I always like to take every time that I'm developing an experiment, looking at data, I like to take a look back and better understand the different level of functionality and how all of these different levels interact together. And this is also something that has been driven a lot by the Air Force Office of Scientific Research in terms of scalability in our research and incorporating all of these different ideas. So we're looking down, starting at kind of more of the cell membrane layer, moving up, looking at ion channels, and different dynamics. And then we start looking at synapses and how the neurons are communicating with each other. And then at the cellular level and cell network level. And then we are looking at different blood-brain barrier levels, and how then we're looking at the full system level, and cognition. So like this is ultimately like how we perceive information and how we process that through our periphery and our central nervous system. Yeah, and then again, this kind of goes the same way with looking at the electron. The electrons moving into full systems and electrical propagation. And so this is one of the sensing. This is by JP Wong and his colleagues that have developed this Spintronic chip and microfluidic chip. And this is what we are currently using for the sensing aspect of this. But I just wanted to just kind of bring this up here as a picture. So this has been developed, and it's also with the nanotechnology. Okay, so then I do have a question here. Sorry, let me pull this up from. Let me see it in the chat. Yeah, so the question is the goal of the effort to understand how the eye captures information from light, then possibly replicate the non-tissue device, or even grow tissue that can replace missing or damaged eye sensors? Yeah, I mean, this is all, thank you for that question. And these are all hopes for us to be gathering this basic science information to develop devices. And so exactly, like really hoping to better understand. So like there's two things in this. One is an augmentation thing. So being able to see like different wavelengths of light or sense different different molecular activities, different quantum sensing that we are not currently humanly able to sense. So that could be, and also for pilots or for other, to be able to just better understand the exposures that people that undergo that may affect their vision. And then for myself, like being in the medical field, like that's definitely a goal of this work for me is to better understand how we can develop, grow tissue, integrate tissue, or either for kind of like a transplant, like plant transplantation from a combination perhaps of a filament that could to help to improve photoreceptors and improve vision, and that kind of thing as well. So yes, thank you for that question. But it is part of where we're starting is to better understand how very specifically, because the thing is like, it's from our preliminary findings and the way that we are stimulating nerves in the eyeball through different wavelengths of light, and also from previous literature, it's a non-linear solution. So our hope is to come up with a function that would be able to predict like at a precision level for each person individually, and that would be our ideal scenario. But we're starting here by doing some basic research to identify that. Okay, so speaking of the basic research, so this is kind of how we set this up. And so this was originally being done with a frog sciatic nerve. And this is in a Faraday cave with an electrophysiology setup. But what we did was like kind of some of our first runs is we dissected out a eyeball. I have an experience, I randomly kind of have experience dissecting out eyeballs and optic nerves from other work that I've done here at the U of M. And so we started after an animal was euthanized, I dissected out the eyeball and the optic nerve. Not I mean as far as I could at that. This is for some of the preliminary data here. And then we used a stimulant. We stimulated. Well, this setup I'm showing you here. We used a stimulus of looking at using like an electrical stimulus, non-contact electrical stimulus, and laying over the electrodes. So the bipolar configuration. We're looking at our next steps are using needle electrodes, other types of electrodes using just basic electrophysiology types of equipment to be able to stimulate, looking at maybe stimulating a little bit deeper into this was kind of our first run. And then using quantum sensing to look at that downstream output of the nerve. And then capturing that using signal processing and electrophysiology. So next I'm going to show you a video. And so what we ended up doing was we then used this was a kind of our preliminary way of evaluating this, is we used a phone with a known frequency of strobe, and exposed that to the eyeball in a dark room, and then captured the downstream. So we wanted to basically see if we could capture that electrically, and so that is something that we're still kind of trying to work through some of the kinks of that aspect. But okay, I'm sorry. I'm not sure why that's not working. Thank you, Patricia. Okay, so I guess from this photo though, you can see that we have the phone up to the eyeball, and then we expose that to light, and we're able to identify the electromagnetic stimulation there. So the idea here is that we're collecting a lot of data. And so there's a lot of bench work that goes into this from many of the different collaborators. And it's really, we have this big haystack of data, and we have multiple different kinds of data from it. And a huge part of this work is benchtop data, but we also have clinical data, or either exposure data, like other data that we have from human data, I should say, as well. And we have imaging, different kinds of imaging modalities. We have databases, like larger databases that include outcomes, and then we have omics and everything from metabolomics to all the omics, all the exposomics, everything there as well. So it's just it's a huge amount of data. So like when we're doing a lot of this testing and really trying to create this platform, a big thing is really how we can leverage the artificial intelligence and some of these other data analytics for the precision aspect of this. So yeah, so just, a huge part of it is that the artificial intelligence and machine learning. I know this isn't necessarily maybe about that, but just to kind of mention a little bit that using these concepts is really about the idea of prediction and how we can parameterization. So the prediction of how a cellular network or a nerve will react to a certain combination of different electrical magnet, spintronic activation. And so if we know what will happen physiologically, then we have a better idea of how we can create precision medicine in that. And then also bringing in that engineering framework and some of the adaptive technologies. And I'm just going to briefly touch on the concept of how we can incorporate multi-physics modeling into this as well in a way to be able to come up with this as a prediction. So I'm going to kind of breeze through this aspect, but this is a very basic high level concept, but if we can taking some of this data, the data that we have, and using basic supervised learning methodologies and training a network allows us to be able to better understand a function at a level that we can better move into this as well. Also looking at unsupervised learning to really try to take that kind of the needle in the haystack out of what we are really trying to get at in combination with using computer vision and deep learning as well. And so some of this comes from better like the electrical propagation, but also deep learning in terms of integrating all of the different parameters that we are collecting. And in prediction classification. So I do want to, this is maybe seems a little bit, so I don't have any sort of example of neuromorphic engineering frameworks or adaptive frameworks other than some work that I've done in Bear. So I'd like just everybody to suspend their a little bit for a second from the vision aspect and I'd like to concentrate on this methodology that we can use in neuromorphic engineering for signal processing in order to really capture these closed loop systems. So I'm going to give you this little example here of bears. And so the other research that I do a lot is studying hibernating black bears, and I've done that in collaboration with the Minnesota Department of Natural Resources for about 13 years or so, and it's been a really fantastic, and also in collaboration with Medtronic. So what we've done previously is we've implanted cardiac monitors into the bears during hibernation, and collected throughout the winter. We've collected a bunch of data. So how that's worked is we have created this. This is kind of a fun little engineering project here, but we have a car battery, a 50 amp car battery, a timer, and a non-invasive telemetry head that is in the bear's den. And then we have a solar panel and then a cell phone. So during that time, every 4 hours during the winter, the bear, we would get that message back to Medtronic, and that included impedance, ECG, temperature, and we were able to do a lot of data. I mean, it wasn't really like this big data, but it was like a lot of data that helped us to really better understand the physiology of the bears over winter. But cool, so we did a bunch of things about that, like really identifying some of they have over 30 seconds of asystole. Over time, they don't really lose fat. They don't lose muscle over the winter and are able to really have really unique metabolic adaptations when it comes to being able to not eat or drink over the winter and then come out being kind of ready to go and in great physiological condition, and how the adaptations of that, we can better understand that for humans. But in this process, we also we had an opportunity to collect an EEG and an ECG at the same time. And what we wanted to try to do from this was to better understand if we could predict the ECG from the EEG. And by doing that, that's, well, the thought process, which is actually something that's now is being funded for the development of, is adaptive pacing. So more physiological cardiac pacing is long-term much more, it prevents cardiac pacing. You're getting heart failure basically from pacing. And it's a more natural and adaptive pacing strategy to be able to have some of your physiology predict what your heart should be at, instead of that being kind of run by another algorithm. And so what we did is we used a neuromorphic engineering framework from the EEG. We were able to accurately predict the ECG of the bear and train that network. So, okay, I know that this is about bears and not about eyeballs, but this is something that we're trying to use the same methodology in order to create closed loop systems for this as well, and from neuromorphic representation. And so yeah, this is bringing into it. We're also hoping to bring into this a multi-physics aspect. And and in terms of the multi-physics, what does that mean really? Well, we want to be able to simulate. There's a lot of work that's been done that I've been involved with in terms of predicting electrical physiology and ventricular tachycardia and other cardiac anatomies. And again, this is not, it's a methodology, and it's a methodology that we're hoping to apply. But we've demonstrated by looking at anatomy, physiology, and being able to cellular modeling, that we have been able to predict electrophysiology, and that we're able to predict how that electrophysiology, how electrical propagation from different physiological properties actually occur. So that's something that we have, and this is in collaboration with the Barcelona Supercomputing Center, and but it is also something that hoping to do also more collaboration with here at the University of Minnesota, using some of the open source ISLA-RED work. So, but this, yeah, and this is kind of like a little bit going back to the basic science, what we're hoping to do. Can we use electromagnetism, and can we use different activations? Can we have a cell layer in different filaments, and then better understand that from quantum sensing, electrical stimulation, and create a prediction model for a platform that allows us to do this. So, and this is also including this is back to the physiology and electrophysiology. So it also kind of just goes a little bit back to how, well, I like to think of things as like I'm more of kind of like a top-down person rather than a bottom-up person in general. So this has also been a very interesting way to think about things from a bottom-up level. And I truly believe that incorporating both, like kind of thinking of things from parameter estimation from a quantum level to the bedside, and the bedside to the quantum level, and how we can better understand mechanism and treatment strategies is really important. So, yeah, and then also just like combining this with running these computational models, and also with experiments, and capturing this from databases. And then how do we get to the tissue device interface? So I think of it in terms of like, we have this human specific modeling where maybe we're creating, there's a known physiology of a human, and we're creating a model about that known physiology. But there's also something around, this human like me, or an avatar aspect, where we have a group of we have data from a group of people, and then we're trying to predict the likelihood of one person being apt to those parameters. And then also capturing this high performance computing, either with multi-physics modeling or integrating the omics with other types of data and outcomes. And so that we are really hoping to do this and bring this all together for this neuromorphic platform, bringing in the expertise from many different people. And again, I just can't, I can't say enough how much like this as an from an international collaboration has been really, really critical. Had the chance to go to the working group meeting at the Italian embassy in Washington, DC last year, and working with these collaborators, and I've just been really learning a lot, and really hope to integrate this in terms of like the physiology platform. And I also have my two best collaborators up here, Karis and Lily, who are my daughters, and they were at the Institute of Engineering and Medicine Innovation Week this last week, and they have been very helpful. So yeah, thank you all, and thank you to the group, and thank you so much for having me. So
thank you.
Yeah.
Any questions? Any questions from people on the Zoom?
When you're doing tests on the eyeball, do you have to maintain blood perfusion to keep the tissue alive?
Oh, that's a very good question. So I right now we're keeping it in like a PBS solution, but there's not really so like the way that we have things set up right now, like it's like from the cardiac standpoint, like in my background, like I've always been very much like we're perfusing in order to have the correct biological. So that's my immediate thought in that as well. But this has not really been something we've been able to do, but it's very, very much like I'm dissecting it out, and then it's like it's like an hour, you know, like within an hour where we have to be finished with testing. But I recognize that there will need to be some validation and verification that goes into ensuring that those parameters are not an artifact of that it being out of the body and everything too. So I'm definitely thinking about that in terms of how we can do that more. I don't know, I don't know if we can really do that in vivo, but at least to somehow validate it. Does that make sense?
Yes. So if there were a way to maintain perfusion, which would be very difficult, it would be better, because then you wouldn't have, you would be supplying oxygen to the tissue, and you wouldn't possibly be capturing artifacts of lack of blood flow.
Yeah. No, I completely agree. If you have any ideas, I'm definitely open for suggestions. I was thinking potentially like to try to well, I can't get the whole pig in a Faraday cage, but I was kind of thinking along the lines of like some sort of sensing that we could do.
Okay, I have a colleague who has done an arm blood perfusion of an arm, and like a monkey arm and a pig face. So I don't know if that could be adapted, but maybe I'll talk to him about that and see if this could possibly be adapted to an eye.
Oh, that would be wonderful. Thank you. Yeah, I would, that would be fantastic.
Daniel, I see your hands up.
Excellent. Am I audible?
Yes.
Excellent. Question for you, the professor, on the eyeball, you were setting up the lab where you were as projecting illumination into the eye. How were you determining what the response of the retinal cells? How did you measure that? What was your method of picking up I guess the readings from those cells?
Yeah, so that's a really good question. That's what we're really trying to hope to integrate. So like all these different platforms, we're trying to like figure out how we can like put it all together. So basically, like for the start of it, like we're not really like this part of the thing is more like a larger model. So we're not really looking at that as at a cellular level. But what we are looking at we were activating the retina, and we're electrically activating the cells. Those are the two aspects of that experiment. And then we were sensing using a quantum spintronic array to be able to look at the electrical magnetic activity and response from the optic nerve. But I think like in the future, oh, sorry, go ahead, sorry.
No, so you're looking at the optic nerve. I understand that the nerve would have actually several conduits in it for transmission to the brain. Were you differentiating that or not yet at that level?
Not yet, but that's the hope. So like what we were and also like this is again one of those things, like if you have any recommendations, like definitely open to, you know, collaboration or good suggestions or something like, but so we were thinking about trying to like, well, break it down into sensing from the interface of the photoreceptors to the optic nerve and capturing there, and then electrically, I mean, and then also stimulating from there. We had tried to, but yeah, or like we were thinking about trying to like demyelinate. I don't know, like we had looked up to some other possible ways to demyelinate the optic nerve and to try to isolate that better. So this is a kind, I mean, it is, I guess it's early in this aspect of it, the eyeball part is, that's kind of like the early stages. The rest of the platforms are pretty, are like have been published and have been working on. So this is the novel part of trying to put this all together. But I think it's going to take a little bit of parameterization for us to make sure that we're collecting what we think we're collecting and the signals are real, and doing some more of that kind of groundwork, I think. But do you have any suggestions for like demyelination or how to isolate those bundles?
Well, it seems to me that there's been a lot of work done over the last couple of decades on the response of eyeballs from various species, cats and so forth.
Yeah.
And I would think that there would be a lot of laboratory techniques that can be gleaned or copied from the existing body of research. Have you had an opportunity to search that?
Yeah, and also one of the PhD students in electrical engineering has done a pretty full lit research on that, and really where we got like the basis for this, and then the novel aspect of this being the sensing techniques that we're trying to deploy in this. But no, I mean, there's a couple, you know, from the literature, like a couple different ways to look at that, but it's again, like we're trying to really kind of think through like how it is that we can capture what we would like to capture for the closed loop system. And also, I mean, so like, because long-term, what we'd like to do is if we can use, so like within the Cho's lab in Italy in Tor Vergata, if we can print, this has already been done, printing the biologics on a filament that could be used as contact, or as, you know, like for the eyeball. Like if we're how we could test those different combinations of filaments and materials, and in a way that really highlights that we make sure that we're understanding like the mechanism as well. So
there also might be some synergy that could be achieved with Tesla's Neuralink, or the Neuralink people, because they're very good at getting very small electrodes. Now they're really focusing those electrodes into the brain area, but I think it might be expandable to being connected to the optic nerve. It's a much simpler environment than the entire inside the skull. Certainly much more accessible, but that same mechanical technologies that they're using, very, very fine electrodes, I think might give you some ability to capture the signals that you're looking for without having to, you know, I think your requirements are much more relaxed than having to be inside a skull. And I think, and I think they would be amenable to working with you because it does expand the capabilities of their technology as well. Is that a possibility?
I would have to, I guess I would have to check with the group. I mean, I think so. But I will have, yeah, definitely look into that. That would be great, because yeah, if like, you're right, I mean, the parameters for going inside of a skull versus eyeball or optic nerve would be much more relaxed. So that would be ideal to be able to collaborate on that technology and and that's already established too, so.
Well, I wouldn't say it's established, but at least they have techniques, and they probably have the hardware. They reproduce the
Yeah.
hardware and might make it accessible to you, so you don't have to redevelop that.
Yeah,
It does require surgical. They do take advantage of people who are brain surgeons capable, but even those people have many difficulties getting the electrodes in the right place. But I think that's a situation where you wouldn't have so much trouble because you're not so encumbered, and it might even be possible that you don't have to necessarily be able to identify which particular optic nerve which part of the nerve you're working on,
yeah,
because you could determine that from the signals that you're applying to the eye, and then whatever nerves are activated, then you can start establishing a map, and I think that might expedite your research.
that's great. That's great suggestion. Thank you. I will definitely look into that. Appreciate that. It's a good idea.
Any questions from the room? Online people, any more questions? So thank you again.
Thank
It was a really interesting talk, and forward to your great results.
Yeah, thank you.
Thank you. Bye.